So thanks everybody for joining today. We can go ahead and get started. I just have a few slides before turning over to our speaker. So welcome to the Botanical Safety Consortium webinar series. For those of you that may not know, in the Botanical Safety Consortium, we're working to evaluate the suitability of assays for botanicals as complex mixtures. This is a public-private partnership working to try and advance botanical safety by trying to see what new approach methods we can use to screen for toxicity. These are our current working groups that we have really advancing this mission and goals. And this is an exciting time in the BSc. We have quite a few things coming up, including results coming out. We'll be able to share some of those results at meetings, including SOT. So for folks attending the Society of Toxicology meeting next week, we have a couple different events, including a symposium on the Monday and 2 posters. So please join us if you're going to attend SOT. So just a few quick logistics for today. So everybody is muted except for the speakers. Please use the question box if you have a question. We plan to record the webinar and we'll share with participants after in the next coming days. And we will have some time for questions after the presentation. If you have any issues during the meeting, please email me there. And this just shows what it looks like to ask a question, either if you're on the desktop or on the web. So put your questions there. And with that, that brings me to our speaker. I'm really excited to introduce our speaker today, Dr. Yitong Liu, who has been working at the FDA for the past 10 years as a pharmacognosist in the Center for Food Safety and Applied Nutrition. Her research focuses on the in silico and in vitro evaluation of pharmacokinetics and hepatotoxicity of food-related chemicals, which include botanical constituents and dietary supplements. She's also a subject matter expert for the Office of Dietary Supplements program on regulatory issues related to cannabis, kratom, and new dietary ingredient safety. Prior to joining the FDA, Yitong received a BS in pharmaceutical science and a PhD in pharmacokinetics from China Pharmaceutical University, and she's published 25 peer-reviewed journal articles and 3 book chapters related to toxicity and pharmacology, and is a frequently an invited speaker at the FDA and international conferences. In addition to that, she's been an active member of the Botanical Safety Consortium the last couple years, being a huge help in working to advance our hepatotoxicity group and our ADME working group. So with that, I'll turn over to Yitong. So I will stop sharing, and Yitong will make you a presenter, so you can go ahead. Thank you so much for speaking today.
Thank you very much. Let me see.
Are you still there, Yitong?
Yes, I'm trying to share my slides, and I think I have some connection issues.
I can pull them up real quick as a backup if we need to.
Yes, please.
Okay. Alrighty. I'll pull my screen back up.
Let me see. It's a pretty windy today outside, so I think the network is not very stable.
Okay, can you see the slides now that I have up?
No.
Michelle, can you?
Yes, I can see them.
Yitong, do you have them on your own? Maybe we could follow along together. And you just tell me.
Sure. I think. Okay. So can you still see me or no?
We don't see you, but we can hear you.
Okay. Okay, good. That's why I call you using my phone. I kind of knew that's gonna happen. Okay, so let's open the slides, and can we go to the title page, please?
Yep, that's where we are.
Okay. All right. So thank you very much for the invitation to speak at the BSc, and it's really a pleasure to work with the BSc for the past couple of years. And here I met a lot of very intelligent people, and I'm really, it's my pleasure to work with them. Hopefully we'll figure out something creative and useful for botanical safety assessment. And then so today I'm going to share with you my own work, which is done at the US Food and Drug Administration for the past 5 years. I'm going to talk about the Silico and the in vitro evaluation of biokinetics and hepatotoxicity of botanical constituents. Next slide, please. Here is my disclaimer. This presentation reflects the views of my own and doesn't represent active views and policies. Next slide, please. The botanical use as a traditional medicine and herbal dietary supplements continue to grow. During the COVID-19 pandemic, the global market for herbal supplements was estimated at $10 billion, and the US market alone almost $3 billion in 2020. Next slide, please. So along with the broad use, human toxicities, including liver toxicity, has been reported for botanicals, such as faulty, comfrey, and kratom. However, there is limited data available on the toxic constituents and their biokinetics for these botanicals. Next slide, please. There are some challenges for botanical evaluation. First, botanicals contain complex mixtures of the numerous phytochemicals, and then there are limited in vitro and in vivo studies on these phytochemicals. And a lot of times, structural information are the only data available. The traditional animal testing is time-consuming and resource-intensive. Next slide, please. There's a need to explore the new approach methodologies, names, and then we hope can help us understand the safety profile of botanicals. So in today's presentation, I'm going to talk about a couple in silico and in vitro names. For example, hold on. I can show my screen now, I think.
Great. All right, I'll make you a presenter. Okay, you should be able to now.
Can you see it? All right, this is so sudden. Okay, so I'm on the slide 6. Okay.
Yep.
All right. Oh, okay. Well, let me blow it up a little bit. Okay, so today I'm going to talk about a few names in silico and in vitro methods for quantitative structure activity relationship modeling and physiologically based pharmacokinetic modeling, as well as a couple in vitro assays, such as the cytotoxicity assay, enzyme, and clearance assays. And I'm going to use these in silico and in vitro names, either individually or collaboratively, to explore the safety profiles of botanicals. So I'm going to talk about 4 case studies. And then the first one I'm going to talk is QSAR modeling, which can predict the absorption, metabolism, and the hepatotoxicity of botanical constituents. The second example, I'm going to talk about PBPK modeling, which can predict the plasma and tissue concentrations. The third example, I'm going to talk about in silico and in vitro concordance for the inhibition of liver metabolism enzymes. The last example, I'm going to talk about in vitro to in vivo extrapolation for hepatotoxicity. So now we start with the first example. QSAR modeling predicts absorption, metabolism, and liver toxicity. So in this study, botanicals associated with a clinical liver injury has been found from databases and literature. So then the phytochemical constituents in these botanicals are identified, and then their structures were extracted and put into the QSAR models. So using QSAR models, first, these phytochemicals will be predicted for absorption if they can be absorbed into in the GI tract. Then only the absorbed phytochemicals will be used to predict metabolism, to generate metabolites. And then both the absorbed phytochemicals and the metabolites will be predicted by QSAR models for their potential to induce human liver injuries. So here's the list of the 20 botanicals with the reported liver injuries in humans, such as aloe, black cohosh, forty, kava, kratom. They have been used for various conditions, such as weight loss or anxiety or pain, etc. And then next, among the 20 botanicals, 255 phytochemicals were identified. And then here is a list of their chemical classes. You can see anthraquinones or pure listing alkaloids, catechins, purines, etc. And then so now, the first step is to predict absorption. Among the 255 phytochemicals, 193 of them were predicted to be absorbed. And then the rest of the 62 phytochemicals with poor absorption were eliminated from the following studies. So these 62 phytochemicals were predicted to be not absorbed due to their physical chemical properties, such as their large size and poor solubility or poor permeability. Below, there are two compounds, one from germander, one from skullcap. They are predicted to be not absorbed due to poor solubility or poor permeability. And next step, so for these absorbed 193 phytochemicals, and then they're used to predict their metabolism by QSAR models. So in the QSAR models, there are like 5 major liver enzymes, cytochromes P450s, CYP1A2, 2D6, 2C9, 2C19, and 3A4. All these 5 major CYPs, they're used to predict metabolites. Below is a compound in comfrey, and then this compound is metabolized by CYP3A4 to a hydroxy metabolite. And then next step, both the parent phytochemical and their metabolites were used to predict liver injuries. So 3 QSAR models were used to predict elevated serum levels of ALT, AST, and LDH enzymes. They're all biomarkers for liver injuries in the clinics. So among these phytochemicals and their metabolites, 48 phytochemicals were predicted to potentially cause human liver injuries, such as pure listing alkaloids, terpenoids, and anthraquinones. So among these 48, 7 compounds were toxic to the liver due to parent compound only. 26 were toxic to the liver due to toxic metabolites. And then 15 of the compounds, both the parent compound and the metabolites, were predicted to be toxic to the liver. So to conclude, so for example 1, a tiered approach of evaluating absorption and metabolism before predicting liver toxicity provided physiologically relevance. And then second, QSAR prediction agreed pretty well with available in vitro and in vivo data on phytochemicals, such as pure listing alkaloids and anthraquinones. These in silico findings help reduce new toxic phytochemicals in botanicals, and therefore prioritize further testing. Future efforts may expand the QSAR applicability domain for botanical constituents. So now we go to the second case study. In this case study, I use the PBPK modeling to predict the in vivo profiles of phytochemicals. So in this case study, I'm going to talk about cannabinoids. excuse me. So the 2018 Farm Bill defined hemp, which is the cannabis and the cannabis derivatives, with no more than 2.3% on dry weight basis concentration of THC. So since then, there has been an increased interest in developing consumer products derived from hemp and its constituents. Cannabis contains more than 120 cannabinoids, which accumulate primarily in the female flowers and leaves. Here are some examples of the major cannabinoids in cannabis: CBD, CBG, CBC, CBN, and CBL. So in this case study, first, I established the PBPK models for CBD at different dosing routes and levels in humans. And then I used the in-vivo plasma concentration time profiles to evaluate the established PBPK model for CBD. And then 16 additional cannabinoids were used to establish PBPK models. And then the PBPK model predicts the plasma tissue concentrations of these cannabinoids in humans. The first step is to establish a PBPK model for CBD. In order to establish PBPK models, two types of input parameters were needed. The first one is human physiology. And then so in this case study, I used the physiology for a 30-year-old Caucasian male with a body weight of 75 kilograms. And then the second type of input parameter we need is chemical properties, such as water solubility for CBD, which is derived from the literature. And then other physicochemical properties of CBD, such as the clearance and the unbound fraction to plasma proteins, were predicted by QSAR models. So at the bottom of the slide, you see the in-vivo profiles of CBD. On the left, you see the intravenous infusion of the 20 milligrams of CBD in humans. The black squares represent the clinical data in healthy volunteers. The black line is the predicted profile for CBD. And then on the right, you see the oral dose of 30 milligrams of CBD in healthy human volunteers. Again, the black squares represent real data, and the line represents simulation. So based on visual examination, you can tell the established PBPK model simulated the in-vivo profile pretty well. All the key parameters such as the clearance, volume distribution, the area under the curve, and then the maximum concentration in the plasma, and then the time to reach maximum concentration were all predicted within 2 to 3-fold of the real data. So now we have a confidence for the PBPK model established for CBD. Next step, I'm going to predict the tissue concentrations of CBD in humans, which is impractical to get from healthy volunteers. Here you see the profile. The x-axis is the time, and the y-axis is the concentration in the human body. You see that at the bottom of this figure, you see the red profile, which represents the profile in the plasma, with the maximum concentration around 6 nanomolar after oral administration of 30 milligrams of CBD. At the top of the figure, you see a blue profile, which represents the liver. So the maximum concentration in the liver is around 86 nanomolar, which is more than 10-fold higher than the concentration in the plasma. In between the liver and plasma, you see other organs, such as the green file for the brain, purple profile for the kidney, orange profile for the reproductive organs, brown profile for the heart, and interesting, the black profile, which represents the accumulation of CBD in the fat. This is not surprising, since CBD is highly lipophilic and it accumulates in the fat. Okay, so we have done PBPK modeling for CBD, and then next I applied a similar approach to establish PBPK models for additional 16 cannabinoids, which are showed on the x-axis. And then for the y-axis, you see the maximum concentration of these cannabinoids in human plasma and tissues. So at the bottom, the red dots represent the maximum concentration in the plasma. And then at the top, the blue dots, the blue squares are the maximum concentration in the liver. So similar to CBD, for the rest of the cannabinoids, the tissue concentrations, especially liver, are much higher than the plasma. So to conclude, in this case study, PBPK modeling enabled the prediction of in vivo profiles, including tissue exposures of cannabinoids. And then these, the PBPK, the pharmacokinetic profiles of the 16 cannabinoids were similar to CBD, and the in vivo profiles of CBD agreed pretty well with the clinical data. So although the low exposure was predicted in the plasma, the tissue concentrations, especially the liver, were much higher after oral administration of cannabinoids. So these predicted pharmacokinetic profiles may help to interpret the observed toxicity, such as the liver toxicity of CBD. The next example, I'm going to talk about the in silico and in vitro concordance for the inhibition of liver metabolizing enzymes. So cytochromes P450 enzymes are major metabolizing enzymes for various xenobiotics and endogenous compounds, such as drugs, phytochemicals, and environmental pollutants, as well as the hormones, cholesterol, and bile salts. Therefore, the inhibition of CYP activities could lead to serious drug-drug supplement interactions and other adverse effects. In this case study, I'm going to use anthraquinones as example compounds. These anthraquinones are being used as drugs, traditional Asian medicine, and dairy supplements. They have various pharmacological activities, such as laxative, antioxidant, anti-inflammatory, anti-cancer, and antimicrobial. Sorry, excuse me. So in the middle of the slide, you see the two plants. On top is aloe, which contains anthraquinone. At the bottom, you see 40, that also contains anthraquinone. On the on the right, you see the structures of the two major anthraquinones, imodin and rain. So for this study, I selected 22 anthraquinones, and then I purchased these anthraquinones. And the first step is to use the QSAR modeling to predict their potential to inhibit CYP activity. And then I use in vitro assays to measure the inhibition of these anthraquinones towards the major CYPs. I used both human recombinant enzymes and human liver macrosomes. Then a concordance was evaluated between the QSAR modeling and the in vitro assays. So first, based on the in silico prediction, these 22 anthraquinones were inhibitors for CYP1A2, 2C9, and 3A4, but they're non-inhibitors for 2C19 and 2D6. So now that's the now we move to the human recombinant enzyme assays. Here are the results. So in this table, you see the 22 anthraquinones, and then you see the 5 liver CYP enzymes. You can tell a lot of these anthraquinones are inhibitors for 1A2 and 2C9 in human recombinant enzyme assays. And then for a few anthraquinones, such as imodin or aloe-imodin, they can inhibit multiple CYPs. Only the IC50s under 10 micromolar were listed here in the red boxes. So the green boxes, they're non-inhibitors because their IC50s were about 10 micromolars in human recombinant enzymes. So next step, 5 of the anthraquinones were tested in human liver macrosomes. And then for the same 5 enzymes, for CYP3A4, we use 2 substrates because CYP3A4 has multiple active center for different substrates. We use both midazolam and testosterone as the substrates for CYP3A4 activity. So here you see the in vitro inhibitor concentration of the 5 anthraquinones towards 5 CYP enzymes. And then from these results, you can tell the inhibition in the human liver macrosomes were weaker than in the human recombinant enzymes because the IC50 values were much higher than the human recombinant enzymes, except for CYP1A2. So in both human recombinant enzymes and human liver macrosomes, anthraquinones can strongly inhibit CYP1A2 activity. So the IC50s were under 10 macromolar in both human recombinant enzymes and human liver macrosomes. So these are just in vitro results. So next, I go to the literature. I found the plasma concentrations for the 5 anthraquinones. So here on the left, you see a figure, and then you see the 5 anthraquinones and their plasma concentrations in humans and animals. For example, you see the compound RAIN. The plasma concentration of RAIN spans a really wide range. It could be as low as below 0.01 macromolar. It could be also as high as about 100 macromolar. So this is a really wide range. The same thing for emulsion and alloemulsion. For propylene and rubidium, we didn't really find too much in vivo data, so only a couple concentrations were found in the literature. So on the figure, you also see a couple red boxes. The red boxes correspond to the in vitro IC50s for these 5 compounds. So if you look at the table on the right, for RAIN, the in vitro measured IC50 for CYP enzymes were between 7 macromolar and 79 macromolar. That corresponds to the red box on the right, on the left. So here you can see the in vitro inhibition, the in vitro IC50 values fall into the in vivo plasma concentration range. So that means RAIN, emulsion, alloemulsion could inhibit CYP activities at plasma concentrations. This indicates potential in vivo inhibition could happen. So of course, this needs a further evaluation in vivo. Okay, so to conclude, the in silico prediction and the in vitro human recombinant enzyme assays showed good concordance. 75 to 90% of the anthraquinones tested here were correctly predicted for 1A2, 2C9, 2C19, and 2D6. However, only about 50% were correctly predicted for CYP3A4. This is quite disappointing, but understandable. The CYP3A4 has multiple active sites, and then has the structure for the substrate were really diverse for CYP3A4. So then last, these in silico and in vitro results enable the rapid identification of potential CYP inhibitors and prioritize future more in-depth studies. So the last example, I'm going to talk about the in vitro to in vivo extrapolation for hepatotoxicity. So in order to conduct this in vitro to in vivo extrapolation, we need help from PBPK modeling, which can predict the absorption, distribution, metabolism, and the excretion of a chemical in humans. So here we have in vitro liver toxicity, which could be measured in cell models using in vitro assays. We can generate a dose response curve and then calculate a EC50 value based on in vitro assays. And then next step, we can use PBPK modeling to translate this EC50 from in vitro assays to in vivo hepatotoxicity. So first, we measure in vitro toxicity in cell lines. We generate a EC50 value, and then we set this EC50 value equal to targeted plasma concentration, CSS. Then we use PBPK modeling to reverse translate the CSS to a predicted toxic dose, detox, in humans. And ultimately, this predicted toxic dose could be compared to the estimated daily intake, EDI, in humans, and the ratio could be used to evaluate safety. The first step is to evaluate the in vitro cytotoxicity of 16 anthraquinones. Again, here anthraquinones we use as example compounds. And then for the in vitro cell models, I use the HER2C3 cells and HUH7 cells. So in these two cell models, I use a 72-hour treatment for both single dosing and the repeated dosing for every 24 hours. And at the end of the treatment, ATP was measured to determine cytotoxicity. So on the figure, you can see the distribution of the EC50 values for the 16 anthraquinones. So imodin is the only compound who has the EC50 value under 15 macromolar. And then there were 3 compounds with EC50 values between 15 to 50 macromolars. And then another 3 compounds with EC50 between 50 and 100 macromolar. And then the rest of the anthraquinones had EC50 values about 100 macromolar. And next step is to establish a PBPK model. And then for input parameters, first we use the physiology. Here I chose a 70 kilogram human subject. And then immediate release tablet as the formulation. And then next I put in the in vitro EC50 values. I set this value as a targeted plasma concentration. And then I also input the in vitro measured clearance in primary human hepatocyte. And then the rest of the phytochemical and the PK properties were predicted by QSAR models, such as water solubility, effective human jejunal permeability, and the unbound fraction to human plasma proteins. With all these input parameters, the PBPK model can predict a toxic dose for these anthraquinones. Here are some of the example results. So in this table, you see 2 anthraquinones, imodin and rain. And then first, the EC50 values were listed. For imodin, its EC50 value is around 15 macromolar. And then this 15 macromolar was set as the targeted plasma concentration. So the in vitro measured clearance in primary human hepatocyte was around 100 microliter per minute per million cells. And all these data were put into the PBPK model, together with the water solubility, permeability, and unbound fraction to plasma proteins. Then the PBPK model predicted a toxic dose. For imodin, this toxic dose is about 1,000 milligrams per day, which is the cutoff value for the program. And then I went to the literature. I found the estimated daily intake of imodin. So here I want to clarify, to assess toxicity in a very conservative manner. This estimated daily intake was calculated by multiplying the highest detected levels of each anthraquinone in a botanical by the maximum human daily intake of the botanical, such as most likely in traditional Asian medicines. And then for dietary supplements, this estimated daily intake level is much lower than traditional medicines. So for imodin, I calculated the ratio between the toxic dose and the estimated daily intake. This ratio is above 3.7, which means the toxic dose is much higher than the estimated daily intake, indicating imodin even used at its maximum level in humans, is not likely to cause toxicity. So on the other hand, you see the other compound, rain, on the right. So the in vitro determined EC50 for rain is about 44 micromolar. And this concentration was set to the equal to the targeted plasma concentration. So in the primary human hepatocyte, rain was not metabolized. So the clearance is set to 0 for liver metabolism. And then the water solubility, permeability, and bond fraction to the plasma proteins were predicted by QSAR models. The PK model predicted a toxic dose for rain at 114 milligrams per day. Then the estimated daily intake level for rain is about 429 milligrams per day. Therefore, the ratio between the detox and the EDI is less than 1, which means the toxic dose is lower than the daily intake, which indicating when used at its maximum concentration in humans, potentially one can cause human liver injuries. So to conclude, in this last case study, an integrated approach was used to combine in vitro cytotoxicity and the in silico modeling into the quantitative liver toxicity evaluation for anthraquinones. And the rain was identified as a potential hepatotoxicant due to a combination of cytotoxicity, plasma concentration, and daily intake level. So the in vitro to in vivo extrapolation enabled rapid hazard identification and a compound prioritization for future more in-depth studies. So to summarize the 4 case studies, in silico modeling and the in vitro testing through data gaps in evaluating biokinetics and safety for data-poor botanical constituents. Future efforts is needed to expand the QSAR applicability domain for phytochemicals. And more in silico, in vitro, and in vivo concordant studies are needed to evaluate and improve the performance of NAMs in predicting human effects. And then last, I want to acknowledge some of my colleagues. First, my postdoc fellow, Dr. Sumulu Mapa, for her help. I also want to acknowledge my managers, Dr. Sprendel, Dr. Salamodis, and Dr. Torrance in the Office of Applied Research and Safety Assessment at the Center for Safety and Applied Nutrition at the US FDA. Thank you. And then I'll take questions.
Great, thank you so much for that presentation, really excellent. And I really like the case studies you used to highlight the different tools and how they work together. So if folks have questions, they can put it in that question box. We got one from Rick already in the chat, and the question's related to the in silico tools for absorption and toxicity. So Rick asked, was there an upper limit on the I think his question has to do with when you said things were not absorbed, did that take into account the toxicity, or was it more the absorption? Did that come into play at all? And Rick, if I mischaracterized that, please let me know.
Why predict absorption is mostly based on the physical chemical properties of the compound. It's not, it's not related to toxicity. It's more based on the, some allergies, based on the physical chemical properties, such as the compounds are too large and then has a multiple charge, or has a poor water solubility or poor permeability. It's all based on the physical chemical properties.
Yeah, that's helpful. And that's something, Yitong, we've talked about in the BSc group, how do we incorporate if there's any toxicity first? Do we look at absorption first? So I think some of those discussions kind of field wide are going on to see how, maybe what order we tier them in. Like, do we consider any of the toxicity or the absorption first? So I think that's a good point and things we can continue to think about. And another question.
That's very interesting.
Oh yeah, sorry, go ahead.
Yeah, sure. Yeah, because that's a very good question. So some toxicities can happen in the gut as well. Then that may interfere with your absorption. That's actually a very valid concern. But in this case study, I didn't consider that. But that's something to keep in mind. Some compounds may already have a toxicity in the gut, which may influence the absorption. Yeah.
Mm-hmm. Okay, a question came in from Ed Fletcher and asked, what is the average weight for an adult on which the study was based on?
Yeah, so in this in my case study, I used for the cannabinoids, I used the 75 kilogram human subject. So the reason why I chose 75 seems a little bit low, but this is consistent with the clinical reports in those healthy volunteers. And then so there are like some clinical studies for CBD pharmacokinetics in healthy volunteers. The average body weight for them is 75. I know that doesn't represent the whole population, but that's at least I kind of want to keep consistent with the clinical study.
Yeah, thanks for that. And I think sometimes that's an assumption based into a lot of models as well, right? That like a range of weights. Yeah.
Okay. Right. In the model, you can choose the body weight, and then but I do think in order to predict toxicity in a more conservative manner, we want to kind of use a lighter body weight. So that's kind of with the given dose, the in vivo concentration could be a little bit higher. It's very conservative.
Mm-hmm. Okay, Jay Petrick asked, you showed prediction data for liver enzymes. How did you predict what ALT, AST, etc. would be?
Oh yeah, so that's the you mean for the first case study, right? So for these for the liver enzymes, these these three liver enzymes are commonly used as biomarkers in clinics to diagnose liver toxicity. So there, I use the QSAR models trained using human data on these three enzymes, and then to predict the potential elevated enzyme levels for phytochemicals. Is that what the question is?
I think so. Maybe it was just on what model you used. Did you say that?
The QSAR models, yeah. These are just trained QSAR models, yeah.
Okay. Okay. Barbara Sorkin asked, could you talk a little bit about what's inside the black box of the QSAR model in terms of the data and computational methods?
Oh, well, I mean, I use a commercial program, and then it's developed by Simulations Plus. This program is called ADMET Predictor, and they have a proprietary training data set. And but I mean, from the menu of the program, I see they have a pretty good prediction for drugs. So, but they have their artificial neural network as their training model, and then but that's all I know. I don't really know the training compounds in those models.
Yeah, a lot of times for the different models, they're proprietary, and we may or may not have access to that, or the false positive, false negative rate, things like that. We may or may not have access to.
ADMET Predictor, though.
Right. I mean,
go ahead, Michelle.
Oh, just to jump in. I mean, ADMET Predictor and folks from Simulations Plus have worked with the consortium, so a lot of that information is available. If you go to their site, they have kind of a lot of multiple hundreds of pages of manuals sort of describing it. So it is a commercial software, but a lot of their details of these models are available through their site.
Great, thanks, Michelle.
I mean, for some of the so for a lot of these models were trained by drugs, drug-like compounds. I mean, because for drugs, we know more about them, so it's easier to get a good quality trained QSAR models. That's one of the challenges I have for phytochemicals, because some of these phytochemicals, their structures are not drug-like. And then they have multiple sugars and stuff. So some of these compounds they're outside the applicability domain of those QSAR models. But I think in the future, when we generate more high-quality data for botanicals, and then we can kind of try to improve these QSAR models when we have like high-quality data on botanicals. So some of these QSAR predictions agree pretty well with existing literature reports on some of these botanicals, such as the anthraquinones and the pure living alkaloids. And I think the QSAR models did a reasonably well job for some of these phytochemicals. But for others, they're kind of too far away from the training structures. So those at current stage, we couldn't really do much. But in the future, like I said, once we have the high quality data, we can expand the applicability domain of the QSAR models to make a better prediction.
Yeah, that's a good point. Saramia asked a question about the compound Reane. So she asked, using 0 for clearance in your PBPK model based on lack of metabolism, but doesn't Reane itself get cleared?
It does. I mean, that's a good question. Here, it's just a very brief, overview of the project. So for Reane, yes, it's not cleared in the hepatocyte, but does have a kidney clearance in the urine. So that's considered in the PBPK model, the kidney clearance. It is cleared, yes.
Great, thank you. A couple more questions. Barbara had another question about the SIPs, where there was poor concordance with the in vivo data. Do you think that's a function of the compound studied or the model itself? If it is the model, is there a way to improve the models?
For the SIP study for the inhibition of CYP enzymes?
Yes.
I think so. Hold on. There's actually pretty good concordance between in silico prediction and human recombinant enzymes. That's a very good concordance. 75 to 90% of the anthraquinones were correctly predicted for 4 out of the 5 CYP enzymes. Actually, that's a very good concordance. But for CYP3A4, like I said, because this enzyme has multiple active sites, I mean, it's really hard to make a very accurate prediction, I guess. I kind of understand that, but CYP3A4 has such a high broad substrate diversity, so it's just really hard to predict the inhibition of this enzyme. But for the rest of the compounds, I think the QSAR model did a very good job predicting the inhibition. At least it's consistent with the human recombinant enzyme assay. So we know a lot of times the enzyme assays are more sensitive than the human liver macrosomes, because the macrosomes has multiple enzymes inside. But for the recombinant enzyme, you got only a purified enzyme. There's only one enzyme. It's more sensitive.
Have you ever looked into anything like molecular docking or some of the more advanced models, maybe beyond a QSAR, for maybe the problematic ones like CYP3A4?
I mean, I haven't, sorry, because I don't really have the expertise, but I think that's a very good idea. I think that's something the BSc can consider to do molecular docking. But I think that can definitely be helpful for enzyme like CYP3A4, because it's so large and then just has a really diverse structure for substrates.
Yeah, just a thought, especially if we know, based on your data here, that it's not as good for prediction. Maybe that's in cases where we look for something else or see if there are ways to improve the models.
Yeah.
Okay. Question from Krishna. So really overall, we're looking at the individual constituents here, but what about different phytoconstituents interactions with one another and their impact on hepatotoxicity? Are there any ways to incorporate that into the PBPK models?
That's a excellent question. So that's the one of the challenges we have for in silico modeling. it takes in structures, and then for extract, you have so many compounds, and then you can only evaluate them one by one, and then they may have synergism or antagonism or just additive effects. But we're trying to figure out something. Maybe we can combine them like in silico. But I think it's a pretty difficult job to do. I mean, at the current stage, I don't really have a good solution for that. The only thing I can do is just kind of pick a few major compounds and then take a look at them first. And then, I mean, it's possible for the enzyme inhibitors, maybe you can predict a interaction between compounds using in silico the PBPK modeling. But that's just one or two compounds. You couldn't really do the whole extract. That's a big challenge for in silico modeling.
Yeah, and I think it's something too that depends on each person's scientific question when they use these tools. So if it's something, maybe you have in vivo or in vitro data, but you want to know why something's happening, I think that's a real strength to some of these in silico tools. So I think, there are limitations, and to note those, and I think you stated it well, but to know when to use the tool and maybe when not to, that yes, it won't capture everything, but how can we use these to advance botanical safety, I think is important.
I think a lot of these names, they provide just a piece of the puzzle. You couldn't really use one model to do it all. I mean, let's be kind of realistic. Even in vivo animal studies, couldn't just do it all. I mean, one model, one endpoint, you couldn't just capture the whole thing, the whole picture. We need a multiple of these names to kind of provide information from different angles. Then you piece them together, hopefully we have a better understanding of the botanicals. But I don't think one tool or just even a couple tools can solve the problem.
Okay, question from Olaf Kelber. What is the relevance of metabolism by the gut microbiota in general? Do you have any experience with that?
I mean, actually, that's very important. I mean, that's especially important for the botanicals. A lot of these botanicals have sugars, right? And then glycosides. And then a lot of, sorry, excuse me. These sugars could be removed by the microbiome in the gut. And then, so these glycosides will be metabolized, hydrolyzed by enzymes in the gut, and then they release I-glycone, and then to improve the absorption into the system. So that's actually a very important part of the in vivo profile for botanicals. But in the current stage, we don't really have a model to predict the metabolism by the gut microbiome. At the BSc, we try to manually remove the sugars from the glycosides. And then I think it's one way to do it at the current stage. But that's actually a very important piece of information for the predicting in vivo profile for these botanicals. Very good question.
Okay, the last question I see is a follow-up from Saramia on the earlier question about the compound Rien. So I guess maybe more statement. I wonder if using 0 for hepatic clearance overestimates the toxicity as there are quinone reductase in the liver. Do you have any thoughts on that?
Good question. Good question. Yes, that's actually very good. I mean these. So for this specific study, I used the primary human hepatocytes, and then I don't think it has the all of the enzymes in the liver, and then it only some CYPs. So yes, it could be this model could underestimate the metabolism. Like I said, this is a really conservative evaluation, and then so even if we underestimate the metabolism, the clearance for rain, and then so from a safety assessment point of view, that's we can take that error And then because we'll have to do some follow-up study, like even in vivo animal studies for rain to see if it really actually causes liver toxicity. This is for the purpose of this study is to prioritize and then see among all these anthraquinones, which one we're most concerned of. And then from these results, we see rain maybe the one has a potential to cause liver injury. So we take this compound and maybe we'll do a in vivo study or like some other like a better system, like the organ chip, which kind of claim has like a better metabolic profile, or we can incorporate multiple organs like a liver and kidney, right? Like a fancier in vitro models. Then we can incorporate both liver and kidney clearance for rain. Like I said, this is a very conservative estimate. And then the purpose is to prioritize for future testing. I'm not saying rain is toxic, but I'm just saying, among the 22, rain is the one I'm concerned of, and then we may need more follow-up study.
Mm-hmm. No, I think that's a helpful way to look at it and see what these tools can tell you, and then to see what you would need to do further on.
Right. I mean, there are like so many compounds in botanicals, right? We have to start somewhere. I mean, you prioritize them little by little, from 1,000 compounds to 100 to 10 to like just a couple, and then we have the resources to actually to dive deeper for these couple compounds.
That was all the questions I see, and I think given the time, we can give you a break from questions, and just thank you so much for this presentation. A lot of great learnings I think we can apply to the BSc, and maybe folks can apply to their own research. So just really thank you, and for the audience, continue to look for BSc events, either in person at conferences or these webinar series. So thanks everybody. Thanks Yitong so much. Have a great rest of your day.
Thank you.